Okay, so here with an amazingly beautiful blue Cambridge sky, as we have today, is a picture of the Genome campus, which has global recognition for genomics, omics science, and also data, and is the largest concentration of hubs of computational biology and genomics in Europe. We're very proud of what we have. So today we have Sanger, we have EBI, and as of this month, the new NHS Health Data Research Service will be based at the campus as well. So we have a selection of modern buildings here, as you can see, as well as sequencing labs, as well as the historic campus, so Hingston Hall, and the conference centre as well. So at the Wellcome Sanger Institute, which is most famous for sequencing the human genome, we have ongoing research programs, which I'll talk a little bit more about in a moment, doing single-cell sequencing, somatic genomics, and also the Tree of Life program. So doing very deep research across these modalities. We also have the European Molecular Biology Labs Bioinformatic Institute. It's part of a European 27 member state organisation. It's not to do with the EU, so Brexit has not made any difference to that. And that has several hundred people also based at the Hingston campus. And then, as I said, in July, the Health Data Research Service will join us to provide access to comprehensive NHS health records and research-ready data sets to open up advanced diagnostic data through secure data environments. And we expect that to have a focus on clinical trials data and on pharmacovigilance type activities aligned with the modernisation of the MHRA. So as that becomes clearer during the rest of the year, we'll be really excited to be part of that story. And of course, all three of these organisations are funded to a greater or lesser extent by the Wellcome Trust, who provides long-term stable funding to the Sanger Institute, who contributes to EBI, and also is working hand in glove with government to support the development of the Health Data Research Service. So that's what we have today, but our expansion plans are very ambitious, and here you can see a diagram of what we expect in the next 10 years to be built in terms of the campus. And actually what's really exciting is that this is now underway. You may have noticed, if you live near here or you work near here, that we have 2 big cranes on site, and we're up to the 2nd floor on the first building, which we expect to open in early 2028. So I'm going to talk a bit more now about the science that we have on site at the moment, and then return to some more details about our programme to expand and build. So our heritage on the site is based in the Human Genome Programme. About a third of the Human Genome Programme was developed in the UK, and that was focused on the Sanger Institute. And this heritage carries forward today from genomics through to all the other sorts of omics that many of you will work in and be familiar with, so multi-omics, proteomics, and so on, to really characterize at a deeper level what's going on inside the cells. The second thing that the campus has underpinned is AlphaFold. This capability to understand protein folding was developed based on data held by EBI, the Protein Data Bank. And although the Nobel Prize wasn't awarded to anybody on the campus, actually that data was absolutely critical to allowing AlphaFold to be developed. More recently, the Human Cell Atlas, with its UK node being focused through Sarah Teichmann and Ms. Hanifa, based at the campus, has taken the ability to really deeply profile individual cells, to understand cell state, cell function, and now also to drive more deeply into disease and development, to really characterize the DNA, the RNA, the omics, and the proteins in these cells to understand their function and help then to move forward into digital twin models of them. And it's these digital twins that we think will allow us to build over the next 10 or so years a virtual human. So building models of all of those different steps that help us to understand disease, biology, to understand the functioning of the cells. So all of these capabilities are on the campus, and we're building through them to the future. So at Sanger, on the campus, we have 3 chunks of capability. A focus on disease genomics through the CellGen and through the CellGen program, through the somatic genomics program, where we look at mutations which arise, and then the effect of that disappears and doesn't turn into cancer, and human genetics, where we look at the single gene deletions in particular, which are behind particular human diseases. We also have a focus on planetary genomics through our Tree of Life program, looking to sequence all living things, and we started with a focus on the UK and also with an objective to sequence one organism in each family. We also then have a newer program. Over the last few years, we've developed a program called Generative Genomics, so engineering genomics, thinking about how we build organisms which are not there and refine them for effectively, in the end, for clinical utility. So we believe that these new technologies are transforming research, and we're showing how that's done on the campus. DNA sequencing, moving into single-cell omics, and then incorporating machine learning and AI into the approaches that we use and refining, for example, deciding which sequencing needs to be done using AI. So this gives us a new way to address biology, sequencing species, using AI to design and discover new molecules and test them at scale in tissues and in organoids. In the CellGen program, we have an approach called Lab in the Loop. But what's really cool about this slide is it shows you that in one tissue, you can stack all of these different sequencing modalities to understand the multiomics and the proteins that are represented in an individual cell. So there's many different types of sequencing modality which are that are used to do this, which can build a very, very rich picture that can then be used to develop these models as part of a lab in the loop. So here you can see this concept of a lab in a loop where, for example, you perturb a particular gene that might change the protein which is developed. You can then look at that in vitro and understand the implications, and then use a model in order to determine which perturbation you then want to do next, and to understand what the implications are for cell function, and therefore in disease and potentially in designing treatments. And the richness that this set of technologies and techniques brings together means that you can characterize at a very deep level multiple parts of the human body, and here you can see it in a human embryo. The second program to touch on is our Generative Genomics program, and here we're looking to move from molecular biology to programmable biology, helping us to improve gene therapies, to understand de novo mutations, to accelerate drug development, and in the end, to engineer proteins for use. And this Generative Genomics program allows us to generate data and then to test again in a lab in a loop concept. And we can look at many different features which then help us to design these proteins more specifically. The second organization alongside Sanger is EMBL EBI. And this has the world's biggest set of open access data resources, which is available to organizations all around the world to help accelerate science. It's something we're extremely proud of. The focus of research areas is extremely diverse. It's not just bioscience, but also reaches across agritech, sustainability, microbiomes, evolution, and incorporating both omics data and also spatial data and imaging to build a very rich picture. And this allows EMBL EBI to have a series of very high impact industry collaborations through life sciences and pharma industries, omics, and also into technology. They also collaborate across public and private to support the development of AI models and work with organizations like NVIDIA and Google, where they've announced some very significant partnerships recently. So I'm going to talk a little bit more now about our expansion plans. So here you can see a diagram, and highlighted are the two buildings which are opening in 2028. So at present, we have 125 acres in the campus, and that is focused on science buildings as well as the conference centre, some accommodation for guests attending conferences, but also a nursery and some food and drink facilities. In future, we'll grow to 440 acres and 1.6 million square feet of new laboratories. And this is funded by almost £500 million worth of investment from the Wellcome Trust, in addition to all of the long-term funding for the operations of the organisations on the campus. At the moment, we have around 2,500 people on the campus, and we anticipate that will expand to 9,000. We're going to build a new data centre to enable high-performance compute, as well as integration into the national infrastructure, and there will be up to 1,500 homes for campus workers because we recognise the challenges of affordability in the local area. The Hingston Hall will be redefined as a hotel, expanding the conferencing capability and convening learning and development, and skills, including AI skills, will be a really big part of what we have in the future. We're also opening a health and fitness centre and padel pitches and so on, and building new infrastructure so that you can move from the existing institutes on the south side of the road. If you've got a cursor here. No, you can't see very well. The existing infrastructure on the south side of the road and the new buildings, as you can see, on the north side of the road. So there's going to be two bridges. And we're also thinking about making it more welcome and opening by removing the gates to the campus, although individual buildings will have card access. So really, it's about placemaking for science as a great place to be, cornerstoned on the current globally leading capability, but also bringing in many more organisations, small and large, across bioscience and AI to help create a vibrant campus for the future. The first phase will open in 2028 with new workspaces, both wet and dry, and an incubator, which I'll talk a little bit more about shortly, as well as some accommodation and the health and fitness club. So the new workspaces will be wet and dry, as I said, with an incubator. There will be AI infrastructure and compute. There will be bridges, an energy farm and microgrid. So it's very sustainable in terms of the building, as well as the homes, hospitality, and many more places to eat and drink than we have at Future. So I've got some lovely forward-facing diagrams of how it's going to look. So this is the building. You may have seen it on the hoardings if you've driven past as well. So open spaces, workspaces, meeting rooms, as well as wet labs integrated. And we're looking, as we develop it over the next 18 months, many of you may know Aline, who's also working. Aline, do you want to stand up and wave at everybody? As we help to refine the balance of wet and dry in these buildings to make sure that it addresses the demand of people who are planning to move there. We'll develop our food and drink offer with 5 different places across the campus, large and small, to address all price points and different tastes and requirements. The homes will be focused in the first phase on 2-bedroom accommodation to rent, because that has been identified as the biggest area, so kind of postdoc starting families, as well as the health club, pool, climbing wall, and facilities. And then just to return to this point about an open campus, so there won't be the gates and the swipe card access at the front gate, as you have at the moment, but allowing people to come in to this place, which is welcoming for science. My role at the campus is chairing the Science and Tech Advisory Group, and I'm delighted to have been able to bring together many people, some of whom are active in the local area, so Andy Richards, many of you will know, as well as Ewan and Matt Hurls. It's fantastic that they're joined by Mel Iverson, who is the new Chief Executive of HDRS, as well as Avi Spear, based in Boston, John Marioni, who works for Genentech in San Francisco, and Neelam Patel, who is the chair of the HRA, as well as Rachel McKendry, who works at the Wellcome Trust. So that binds us in very closely to the Wellcome Trust and their future funding plans for science. We're building this integrated research, commercial and residential ecosystem, and really focusing on the future across AI and biology, where we see enormous investment across the US, China, and also the UK to build on the capability that we have at the campus, and one of the most significant expansions in Europe and indeed in the world in this area. In terms of the pillars of our community and the offer, we see trust and the position that the Wellcome Trust has in the open science long-term funding that has been championed by our organisations as being a really key part of what we have. The skills and AI skills and data generation skills across omics and imaging are also really a magnet for organisations to come to us. About 200 people become available every year on the campus with these very high quality skills, not least because at EMBL as a European organisation, they have a mandate only to employ people for a relatively short period of time before they're then set free to go elsewhere. The conferencing facilities are very good. So we have a lecture theatre for more than 300 people, and we plan to expand our lecture theatres and visiting speakers. And so the scale and capabilities that we have on the campus are really exciting. And so thinking through what we have in terms of a value proposition to different organisations, from our data collection capabilities in genomics and omics, in imaging, and acquiring data, as EMBL and HDRS will do, from clinical and scientific organisations to bring them together, to curate that data and make it available to different organisations, which will then allow it to be used for AI and turned into foundation models. And then finally, hopefully translated into tools, techniques, diagnostics, therapeutics, and approaches for clinical medicine that can be used to advance human health, improve sustainability, and improve agriculture. So as part of what we have in future, we see pillars of advancing automated data generation, advancing AI, and driving greater levels of incubation and translation on the campus. At the campus, the Sequence will be our incubator, and we announced last week that it will be run by Biolabs, a US-based organisation who have, and this will be their first foray into the UK. They have multiple incubators around the world, and they're very experienced in understanding how to support early-stage SMEs, very, very small organisations, as they start out on their path to growth, bringing in additional investors and helping them through those early stages. So Biolabs will provide a range of services, equipment, concierge operational services, and curate events in order to benefit startups who are based in the Sequence incubator on our campus, as well as forming collaborative partnerships throughout the local Cambridge ecosystem, and collaborate with the different functions across the Genome campus, supporting access to R&D infrastructure, bringing in connections to industry and other VCs, and curating the community across academic and research activities. So we're really super excited about these developments. Aline and I would love to talk to you about this later, and I will leave it there for today. Thank you very much for your attention.